Good morning everyone. I am Sumati, working as a lecturer in Government Junior College at Bapatla. I am teaching microbiology subject. Today I want to discuss about Unit 6. Important pathogens. Today we are newly discussing about Salmonella typhi. Salmonella typhi. Salmonella typhi. This Enterobacteriaceae, it belongs to Enterobacteriaceae family. Enterobacteriaceae family. The genus Salmonella contains mainly two organisms. Salmonella, the genus Salmonella contains two organisms. One is Salmonella typhi and the second one is Salmonella paratyphi. సాల్మొనెల్లా టైఫీ అనేది ఎంట్రోబాక్టీరియాసి ఫ్యామిలీకి చెందినది. అలాగే దాని సాల్మొనెల్లా జాతిలో రెండు రకాలైనటువంటి ప్రజాతులు ఉన్నాయి. ఒకటి సాల్మొనెల్లా టైఫీ మరియు సాల్మొనెల్లా పారాటైఫీ. ద జీనస్ సాల్మొనెల్లా కంటైన్స్ two organisms or two species. One is the Salmonella typhi and another one is the Salmonella paratyphi. It is the commensal of intestine. Salmonella typhi is the commensal of intestine. Intestine లో ఉండేటువంటి బ్యాక్టీరియా and it causes the typhoid fever. Typhoid fever. Salmonella typhi causes the typhoid fever and Salmonella paratyphi causes the paratyphoid fever. Paratyphoid fever. Salmonella typhi causes the typhoid fever and Salmonella paratyphi causes the paratyphoid fever. And it contains, the genus Salmonella contains two groups. One is enteric fever group and the second one is food poisoning group. The genus Salmonella contains two groups of bacteria. One is the enteric group and another one is the food poisoning group. Next one about morphology. Morphology. Salmonella typhi is the gram-negative rod-shaped bacteria. Motile, non-sporing, non-capsulated bacteria. Morphology, Salmonella typhi morphology, it is the gram-negative bacteria and rod-shaped bacilli and it is the motile with peritrichous flagella. Peritrichous flagella. It contains peritrichous flagella. It is a non-sporing and non-capsulated. It look like this appearance. These are the peritrichous flagella. Peritrichous flagella, rod-shaped bacteria with peritrichous flagella. Peritrichous means the flagella present whole bacterial body. Next one, cultural characteristics. Next one, cultural characteristics of Salmonella typhi. Cultural characteristics. It is aerobic bacteria or facultative anaerobic. Facultative anaerobic bacteria. Aerobic or facultative anaerobic. Optimum temperature at 37 degrees centigrade and pH 4 to 7. 4 to 7. And it grows on nutrient broth. It grows on nutrient broth, blood agar, deoxy chocolate agar, deoxy chocolate agar medium, Wilson and Bismuth, Carry-Blair medium. Carry-Blair medium. Salmonella typhi is the aerobic or facultative anaerobic bacteria. It grows at the temperature 37 degrees centigrade and pH ranges from 4 to 7, and it grows on normal media like nutrient broth. When it grows on the nutrient broth, it gives the turbidity. It gives the turbidity. And it grows on the blood agar medium. It gives the hemolysis. Hemolysis. Next, it grows on the deoxy chocolate agar medium. Wilson and Bismuth and Carry-Blair mediums are the special medias for the Salmonella typhi. These are the special medias and it acts as the transport medium. Carry-Blair medium also acts as transport medium for the Salmonella typhi. Salmonella typhi. Next, biochemical reactions. Next, biochemical reactions. It ferments the sugars like maltose, glucose, mannitol. It produces acid but no gas. It produces acid but no gas. Salmonella typhi ferments the sugars like maltose, glucose, mannitol. It produces acid but no gas. This is the special property or special biochemical reaction of Salmonella typhi. It produces only acid but no gas. It distinguishes between other Enterobacteriaceae family. It distinguishes Salmonella typhi from other Enterobacteriaceae family. Next, methyl red positive, Vogus Posca test negative. It gives the methyl red test positive, Vogus Posca test negative. Resistance. Next one is resistance. Salmonella typhi the Salmonella typhi heating at 60 degrees centigrade for 15 to 30 minutes. Also live. These are resistance for at 60 degrees centigrade for 15 to 30 minutes by autoclaving. ఆటోక్లేవింగ్ చేయడం వల్ల కూడా ఇవి రెసిస్టెన్స్గానే ఈ టెంపరేచర్ దగ్గర 15 to 30 minutes రెసిస్టెంట్గానే ఉంటాయి. నెక్స్ట్, యాంటీజెనిక్ స్ట్రక్చర్. Antigenic structure. Now we are discussing about the antigenic structure of Salmonella typhi. It contains 3 antigens: O, H, Vi. O is somatic antigen, H is flagelllar antigen, variola antigen. This is also cytotoxin. It mainly it contains 3 types of antigen. O is somatic antigen, H is flagellar antigen, VH variola antigen. It acts like cytotoxin. The virulence of the Salmonella typhi mainly based on the antigenic structure. Next one, pathogenesis. Salmonella typhi main causes 3 types of diseases. 3 types of diseases. First one is enteric fever, gastroenteritis, third one is septicemia. Enteric fever is also known as typhoid fever. The symptoms of typhoid fever is a 24 hours fever with shivering. With 24 గంటలు చలితో కూడినటువంటి జ్వరం అనేది వస్తుంది టైఫాయిడ్ ఫీవర్లో. నెక్స్ట్ గ్యాస్ట్రోఎంటరైటిస్. ఇట్ ఇన్క్లూడ్స్ ద అల్సర్స్ ఆఫ్ ఇంటెస్టైనల్ మ్యూకోజా. It invades the membrane layer of the intestine. It enters into the epithelial of the intestine and it causes the ulcers of intestinal mucosa. Last one is septicemia. It enters into the bloodstream and cell division and proliferation occurs in the blood. That is called as septicemia. Salmonella typhi mainly causes the 3 types of diseases. One is the enteric fever, that is the typhoid fever. Next one is gastroenteritis. It includes the ulcers of intestinal mucosa, and the last one is septicemia. It contaminates the blood, is called as septicemia, contamination of blood. Likewise, Salmonella paratyphi also causes paratyphoid fever. It also causes the paratyphoid fever. This is the most important disease, typhoid fever. It is very regular in human beings. Next one, lab diagnosis. First one, culture. When in the case of typhoid fever, blood is collected as a specimen. Typhoid fever cases లో బ్లడ్ అనేదాన్ని మనం ఇక్కడ స్పెసిమన్ గా తీసుకుంటాము. అలాగే యూరిన్, అలాగే ఇన్ కేస్ ఆఫ్ ఫుడ్ కంటామినేషన్ ఆర్ ఫుడ్ పాయిజనింగ్, రెసిడ్యువల్ ఫుడ్. These are the specimens collected for the lab diagnosis of Salmonella typhi. Next one, microscope. When we see under the microscope, Salmonella typhi is the gram-negative rod bacilli and it contains the peritrichous flagella with antigenic structure of HO and VA antigens. It indicates that it is the presence of Salmonella typhi. Next, important serological test. Is the Widal test. Vital test is the most important one and it was used regularly for the diagnosis of Salmonella typhi. The vital test is a agglutination test. It is the agglutination test. It is used to detect the agglutinins like or antigens. Agglutinins are antigens H and O. O and H యాంటిజెన్స్ అనే దాన్ని డిటెక్ట్ చేయడానికి మనం వైటల్ టెస్ట్ అనే దాన్ని యూజ్ చేస్తాము. ఇట్ ఈస్ ద ఎగ్లూటినేషన్ టెస్ట్. ఇట్ ఈజ్ మెయిన్లీ బేస్డ్ ఆన్ ద క్లంపింగ్ ఆఫ్ యాంటిజెన్స్ అండ్ యాంటీబాడీస్. ఇది యాంటిజెన్ యాంటీబాడీ క్లంపింగ్ మీద డిపెండ్ అయ్యే ఉండేటువంటి టెస్ట్ అనమాట. ఇది అనేది వైటల్ టెస్ట్ వచ్చేసి 2 టైప్స్. ఇట్ ఈస్ ద 2 టైప్స్, స్లైడ్ టెస్ట్, టెస్ట్ ట్యూబ్ టెస్ట్. Vital test is performed in 2 types. One is the slide test and another one is test tube లేదా tube test. First one is slide test. In the slide test, this is the slide test. Slide test is it is a rapid and regular test. Regular గా మనం చేసేటువంటి టెస్ట్ స్లైడ్ టెస్ట్. In this slide test there are 4 antigens are taken, OH, AH and BH, and one drop of blood or serum added to these antigens and clumping are observed, and the result scored from 0 to 4%. 0 to 4%. 0 means no agglutination. 1 25%. 2 1 plus 2 plus 50%. 3 plus 75%. 4 plus 100%. 100%. The vital test report or result scored from 0 to 4%. 0 means no agglutination occurs, there is no bacteria. 1 plus means only 25% bacteria present. 2% means 50% bacteria. 3% means 75%. 4% means 100%. Total bacteria is present and it confirms the typhoid fever. Next, tube method. Tube method confirms the results of slide method. This is the confirmation test. Which results are taken from the slide test? It used for the confirmation of the results of slide test. The serial dilutions are taken likewise. Antigen and antibody suspensions are taken these 6 test tubes and it is incubated in water bath at 37 degrees centigrade for 30 minutes. for 30 minutes. The clumping occurs at the bottom of the test tube, or pellets are formed at the bottom of the test tubes. In these, results are scored likewise. Salmonella typhi O antigen 1/80 positive. Salmonella typhi H antigen 1/80 is positive. Salmonella paratyphi AH 1/40 is negative. Salmonella paratyphi BH 1/40 is negative. 1/40 is negatives. In the tube method is used for the confirm the results of slide method. This is also considered as a confirmation test, and the results are scored likewise. Salmonella typhi O antigen 1/80 indicates the positive. Salmonella typhi H antigen 1/80 also indicates the positive. Salmonella paratyphi AH 1/40 indicates the negative, and Salmonella paratyphi BH indicates the 1/40 indicates the negative. Next one, Shigella. Shigella. The scientist named Shiga. The scientist named Shiga first identified and isolated the Shigella bacteria. That's why it is called as Shigella. Shigella bacteria first time scientist అనేటువంటి సైంటిస్ట్ షిగా అనేటువంటి సైంటిస్ట్ ఐసోలేట్ చేయడం వల్ల దీన్ని షిగెల్లా అంటారు. ఇట్ కాజెస్ ద బ్యాసిల్లరీ డిసెంట్రీ. ఇట్ కాజెస్ ద బ్యాసిల్లరీ డిసెంట్రీ. షిగెల్లా అనేది బ్యాసిల్లరీ డిసెంట్రీ అనేదాన్ని కలుగజేస్తుంది. First we see about morphology. Likewise, it also gram negative, rod shaped but non-motile, non-sporing, non-capsulated. Shigella morphology is a gram-negative, rod-shaped bacilli. It is a non-motile, non-sporing and non-capsulated bacteria. Next, cultural characteristics. It grows on normal media or ordinary media like nutrient broth. McConkey's agar medium. Shigella grows on nutrient broth and McConkey's agar medium. A thick turbidity is placed at the bottom of the test tube. That when it grows on nutrient broth, turbidity occurs. Next it reduces nitrates to nitrites. Shigella reduces nitrates to nitrites. This is the special characteristics of Shigella, and for this character we identified the Shigella. ఈ నైట్రేట్స్ని నైట్రైట్స్గా రెడ్యూస్ చేయడం అనే ప్రాపర్టీ షిగెల్లాకు మాత్రమే చెందింది. దాన్ని బట్టి మనం షిగెల్లాని ఐడెంటిఫై చేయొచ్చు. నెక్స్ట్, ఇట్ గివ్స్ ఆక్సిడేజ్ పాజిటివ్ టెస్ట్, catalase also positive. Oxidase and catalase gives the positive taste. It contains 3 types of toxins. It releases 3 types of toxins. First one, endotoxin. Next, exotoxin. Virocytotoxin. Shigella releases 3 types of toxins. First one is the endotoxin, next one is exotoxin, third one is virocytotoxin. This is the endotoxin causes the ulcers of intestine and causes the dysentery. ఈ ఎండోటాక్సిన్ అనేది ఇంటెస్టైనల్ మికోసా అల్సర్స్ని కలుగు చేస్తుంది, అలాగే డిసెంట్రీకి కారణమైంది. ఎగ్జోటాక్సిన్, దిస్ ఈజ్ ద న్యూరోటాక్సిన్ అండ్ ఎంటెరోటాక్సిన్. It acts on nerves, that's why it is called as neurotoxin. Exotoxin acts as neurotoxin, it acts on nerves and muscles. Next one, virocytotoxin. This acts on verocells, that's why it is called as virocytotoxin. Next, pathogenesis. Pathogenesis. Shigella causes the bacillary dysentery. Bacillary dysentery. It is invasive of the intestinal mucosa of large intestinal. It reaches the epithelial cells of the intestinal mucosa and there it causes the ulcers and it proliferation and cell division also occurs there. It releases the toxins and these toxins are responsible for the bacillary dysentery. Symptoms, watery motions and vomitings, dehydration of the body. The symptoms of dysentery is dehydration of the body, motions, watery motions and vomitings. Lab diagnosis. When we see about the lab diagnosis of Shigella, here sample is stool. The stool is the best sample for the diagnosis of Shigella. And next, microscope. When we see under the microscope, Shigella is gram-negative rod-shaped bacteria. And next, cultural characteristics. It reduces nitrates to nitrites. This test gives the confirmation of Shigella. Next, Vibrio cholera. Morphology. It is a gram-negative bacteria, rod-shaped, motile with monotrichous flagella, monotrichous flagella, non-sporing bacteria, non-capsulated bacteria. Non-sporing and non-capsulated. When we see about the morphology of Vibrio cholera, it is the gram-negative bacteria, rod-shaped bacilli, motile with monotrichous flagella, non-sporing and non-capsulated bacteria. Next, Cultural characteristics. Now we are discussing about the cultural characteristics of the Vibrio cholera. It grows on ordinary media. It grows on ordinary media like nutrient broth, alkaline peptone water, .5% NaCl facilitates the growth of Vibrio cholera. We are seeing about the cultural characteristics of Vibrio cholera. It grows on ordinary media like nutrient broth, alkaline peptone water, and 0.5% NaCl facilitates the growth of Vibrio cholera. There are special media for the growth and transport of Vibrio cholera. Venkatraman Ramakrishnan medium. This is the very important medium, and this is special for the Vibrio cholera. Venkatraman and Ramakrishnan medium. Ramakrishnan medium, VR medium. Next, Mansour's telluride agar medium, and Mansour's telluride gelatin medium. These are the special medias which are facilitates the growth of the Vibrio cholera. Next, pathogens. Vibrio cholera causes the disease known as cholera. This is the pandemic disease. Pandemic means affects at a time at a number of people known as pandemic disease. ఒకే టైంలో ఎక్కువ మంది ఎఫెక్ట్ అయ్యేటువంటి డిసీజ్ని మనం ఇక్కడ పాండమిక్ డిసీజ్ అంటాం. దిస్ Vibrio cholera produces the chlorogen toxin. This toxin is responsible for the clinical manifestations of cholera, and it contains the oligopolysaccharide molecule. It is also responsible for the clinical manifestations of Vibrio cholera. Next, lab diagnosis. Stool is the best sample for the diagnosis of cholera, and it transport to Venkatraman Ramakrishnan medium, and the colonies are round, small, and mainly comma shaped. Comma shaped bacteria is Vibrio cholera. This comma shaped indicates the presence of Vibrio cholera.